Welcome to Tackling Antimicrobial Resistance, a new NIHR podcast series that aims to shine a light on the threat posed by antimicrobial resistance to UK and global healthcare and the role research can play in tackling that threat. I'm William Hope. I'm the Dame Sally Davies Chair of AMR Research at the University of Liverpool and also the co-lead for infection at NIHR. Each week I'll be talking to experts in antimicrobial resistance to get their views on what can and should be done to tackle AMR as the NIHR brings together stakeholders to create a UK-wide approach. Hello, everybody. It is my very great pleasure to welcome Professor Neil Gow today. Neil is a Deputy Vice-Chancellor for Research and Impact at the University of Exeter, also Professor of Microbiology and the founding co-director of the MRC Centre for Medical Mycology at the University of Exeter. So welcome, Neil. Thank you very much.
Great to join you, William.
So Neil, I thought we'd start by having you tell us about your career, your very distinguished career, and maybe weaving into some of that history, some of your key achievements and interests in medical mycology.
Well, William, I've had quite a linear career in one sense that I've always been fascinated by fungal systems, right from my undergraduate days at Edinburgh. I went on to do a PhD working on Candida, and that was at Aberdeen with Graham Goody. And that really set the direction of my career right until the current day. I did spend some time in the United States as a postdoc, and I've really loved doing lab science myself. So I had as many sabbaticals as I could squeeze in and legitimately get away with to go back to the lab and do some more work myself. So I've worked a couple of times at MIT and in California and also in Iowa with various key figures in the field. And I've really enjoyed sort of refreshing my own research skills by going back to the bench away from my lab for short periods of time to do that. And then at Aberdeen, we established a significant number of a group of people coming together. Now it's called the Aberdeen Fungal Group. And that was great fun. And we were able to achieve a somewhat of a critical mass of people then. And then I moved to a position at Exeter, which allowed us to actually move the group and actually expand it to further horizons as part of what's now called the MRC Centre for Medical Mycology. And that is an even larger group of around about 100 individuals who study fungal diseases from a variety of angles. And we've got a great network of colleagues which extend out across the country and across the world to other parts of the world, including South Africa. We're setting up a new hub in South America, and the ambitions to have a third one in Southeast Asia. And this creates a group of people working together. I'm mentioning that because actually I think, as you well know yourself, William, that medical mycology is an incredibly important field, but it's not a big field in terms of the number of people who are engaging in it. And we tend to be rather dispersed. And areas where there's a critical mass in one place are rather unusual, but also very helpful for the field in terms of developing skills and creating networks and pooling resources so you enjoy the benefits of critical mass. And I think that's something I've really tried to do in my career. I really passionately believe in that, and I've had the honor of serving as presidents of 4 microbiological and mycological societies, which also have had that role. So my career has really developed in a collaborative environment. I'm a specialist in my own research terms in the molecular cell biology of fungal pathogens, particularly Candida. And within that, I'm particularly fascinated in the walls of fungi because they end up having very important roles in AMR, but many other areas as well, including immune detection, shape, morphogenesis, drugability, etc. So that's a kind of potted history of how I got to where I am. William, it doesn't seem like it was only just a few days ago that I was leaving my PhD to start off as an independent, but it's amazing how quickly you could put decades behind you.
Yeah, I understand that. Neil, just before we get into the beauty that is fungi, and you and I are going to make the case for for this to the world in the next 45 minutes or so. I'm interested in the new leadership role you took at Exeter as Deputy Vice-Chancellor. Has that improved your science, do you think?
Well, it's been extremely exciting, both in terms of the role itself, which is to guide research and impact across the university, and that means all of our current faculties, which includes humanities, social sciences, and STEM subjects. So it's a very demanding but interesting role if you're naturally curious about research from multiple angles. Your question was whether it influenced my own science, and it really has. So, for example, I have started up collaborations with recently with biophysicists and developing new types of biosensors to study in the context of diagnostics for fungal research. I'm using increasingly a range of technologies which I wouldn't have even known about previously that can be applied in the context of our discipline, and it gives it a brand new shape and a brand new set of possibilities. So yes, I've been very much inspired by my new environment, and it's allowed my research to evolve in surprising and different ways.
Right. Now, Neil, I don't know about you, but sometimes I feel in maybe social circumstances, it's a little socially unacceptable to be interested in fungi. They have connotations that are sometimes unpleasant, like mold grouting on bathrooms and toenails, for example. So that both of us know that that's about as far away from the truth as things can be. So why don't you tell us all what a fungus actually is?
Well, I absolutely agree with you, William. And anecdotally, when I'm speaking public engagement events, I often start by showing a little clip from Ghostbusters, where they're trying to establish that one of the Ghostbusters scientists is an absolute nerd, and the sort of one-liner that they used to put down this guy. Egon Spengler his hobby was that he collected spores, molds, and fungus. Presumably, what Hollywood thought was possibly the most boring thing that anybody could possibly do. But actually, at the end of my presentation, I wanted to tell everybody that they've used a fungal product every day of their life, depend on fungi for almost everything that they do, and to make them aware of a group of organisms which are extraordinarily important, and in our context also potentially very dangerous, but are a rich source of excitement which people just don't understand. So for example, let's just remind ourselves that there are about 400,000 plants that are known in the world, but we know that there must be at least 5 million species of fungi. So we've got 10 times the number of fungi than all the plants you can see round about you. So there's a huge biodiversity to think about. That's probably, in terms of total animal species, don't really know how many insects there are in the world, but maybe 1 to 7 million. So it's possible that the fungi are the most biodiverse group on the planet Earth. And within the fungi, let's remember that there's been a whole series of Nobel Prizes which have been awarded to people who study fungal systems, everything from understanding sort of genetics to cell biology. And we are absolutely dependent on fungi for food sources, for drinks, for fermented foods, but also for drugs, for antibiotics, for medically important drugs, for example, cholesterol lowering drugs. They're used in agriculture, so they're really extraordinarily important. And Alexander Fleming was the first of the mycologists to get a Nobel Prize, but Nobel Prizes which have been awarded apparently for cancer, when you look at it in detail, people like Paul Nurse and Lee Hartwell, who got the Nobel Prize for cancer in 2001, actually only studied fungal cells, yeast cells. But from everything from understanding that genes generate enzymes to understanding the traffic and cellular biology of higher cells, fungi have been extraordinarily important. So I am a passionate mycophile and advocate for mycology in all senses. And we know, William, also, although fungi do cause really concerning and difficult to treat diseases, really difficult diseases, which are greatly understudied, despite that, under the microscope, these are aesthetically extraordinary organisms to work with. They're beautiful, they're shape-shifting, they can adopt all series of morphologies, and there's always an exciting study which is being published almost every week in the mycological world. So a really, really important group of organisms.
Yes, I think that they are beautiful, and again, they're not beautiful to scrub off a wall, but under the microscope, like you say, astoundingly beautiful. And so let's just talk about a couple of those things. So you mentioned dimorphism. I think you mentioned dimorphism. All of your work about the cell wall is about dimorphism, Neil. So maybe you could explain that a little bit more about what dimorphism is.
Yeah, so I use that term very broadly. So fungi generally have got two ways in which they reproduce themselves, and they use these two ways of working to generate virtually everything from a simple yeast cell to a very complex mushroom with a very particular architecture. They either form tubes, long filamentous tubes, by growing at one end, or they form spheres, which are like balloon-shaped cells, and they balloon out by growing across their surface. And the difference between those two is where you place your wall. But the way in which, if you regulate that process, you can generate a myriad of shapes and architectures and different cellular structures. So fungi have told us some fundamental things about how things shape themselves and how that's locked into things like the cell cycle, which controls almost everything in cell biology. So the concept of dimorphism is very well explained and exemplified by fungi, and quite easily controlled, because fungi turned out to be, as experimental tools, very amenable to experimental study. We can cause candidates to switch from one shape to another within an hour. So you could take a whole series of yeast cells, rounded cells that look like mini balloons, and they'll form these tubes which extend to form a linear thread. And they'll start to do that process within 45 minutes, and within a few hours, you've got a tangled mass of tubes. So that's a fantastic thing to do. Others do this, some of these do this due to chemical stimuli, others do that as a response to temperature. And in fact, probably the first scientist ever to notice this was Louis Pasteur, who discovered thermally and regulated dimorphism in some of the mucorrhizous fungi. So it's been a model system for a very, very long time.
Can you, for people that won't have heard of these pathogens, name some of the thermally dimorphic fungi? That's the way they're classified, whether they exist as yeast or molds depends on temperature. And these are scattered throughout the world, but maybe, Neil, you could just tell us a little about that.
Yeah, yes, certainly. So I studied a fungus called Candida, Candida albicans. It's important to give the species name because they don't all exhibit dimorphism in the same way, but Candida likes to be filamentous at body temperature. That's not almost enough, but at 37 degrees, it tends to be more filamentous than rounded and yeast-like. Now, most of the other pathogens of the world actually do it the other way around. They tend to be yeast cells in the human body at warm temperatures. They tend to be filaments when we find them in their natural environments, outside there in soils or on the surfaces of things. And that's very interesting. So there's a range of organisms, many of which are restricted for, which we don't fully understand why, but many of these are restricted to particular areas of the world. So if you were a medical mycologist from South America, you would most likely be working on a fungus called Paracoccidioides brasiliensis. The Brazil gives you an idea of its geographical area, but it's not restricted to Brazil. A very interesting fungus, and that transition from the yeast, from the filament to the yeast occurs naturally at 37 in the body. But even more intriguingly, it tends to be suppressed by human estrogen. So women tend to get the disease very much less commonly than men do, more than 10 times, sometimes as much as 80 times less commonly. there are also some yeast-like dimorphic fungi, like Histoplasma capsulatum is another one. Actually has been linked to the death of Carnarvon and those who excavated potentially the tomb of Tutankhamun. It's thought that they inhaled the spores of this fungus, which grows on the guano of bats in the outer parts of the chambers of Tutankhamun's tomb. And you'd probably be aware anecdotally that some of these archaeologists died in surprising and unexplained circumstances. And one suggestion might be that they were a victim of inhaling of the spores of this fungus. But there ends up being quite a number of these dimorphic fungi, some of which are geographically restricted to South America. There's another group in North America, some in Africa. And also there are organisms related to penicillium, which is known as an antibiotic-producing organism, and which are also thermally dimorphic and will convert to yeast-like cells in the human body. So a very interesting trait. And of course, we want to know, is the conversion essential for the disease or not? And that's an interesting set of questions to study in the lab.
Yeah, so Neil, I want to get on to antimicrobial drug resistance and antifungal agents and tell people about that. But there are a few other things just to fill in, I think, just before we get there. One is that fungi have sex, right? And the other one is that they are eukaryotes. So they're closer to humans than they are to bacteria, for example, which are prokaryotes. So maybe it would be just interesting to hear about what the consequences of that genetic relatedness to humans is for antibiotics.
Yes, because the overall concept of an antibiotic is to have a drug, a chemical, which is a silver bullet. It will affect the target that you want to kill, the microorganism, without affecting the human. Now, of course, humans have got metabolism, as do microbes. And because fungi are eukaryotes, that means their biochemistry and their cellular organization is shared with that of human beings. There are fewer natural targets for antibiotics to be raised against. And of course, the fungi are dominated by three main targets. The membrane is interesting and different from human membranes. We have cholesterol in our membranes, and fungi have a different type of sterol called ergosterol. And the pathway to make ergosterol is a very important target for a range of antifungal drugs. Now, the wall is completely different. We don't have a wall. And the wall contains molecules which are almost exclusively fungal in nature. So it's a very rich target for antifungals. But actually, at the moment, there's only a couple of types of classes of drugs, mainly one class of drugs, the echinocandins, which specifically attack one of the important robust polysaccharides which make up the cell wall, without which the fungus is very unhappy indeed and may, in fact, usually will die. So these are two major target points, and there are a few other drugs, but actually overall, the numbers of drugs we have to treat fungi with are many fewer than we have to go out and treat bacteria with. Even more concerning is that if somebody has an infection, a very common and shared symptom will be fever. And to a doctor who hasn't got anything else to help them with, that fever could be, the origin of that fever could be a bacterial infection, or it might be a fungal infection. Of course, it could be a viral infection too. But if you make the assumption it's a bacterium and treat as if it's a bacterium, you'll have no effect whatsoever on the fungus. And tragically, for the treatment of medical mycoses, fungal diseases, late diagnosis nearly always equates to poor prognosis. So the longer you delay using the right type of treatment, the less is the chance of saving that patient. Now, these I'm talking about a set of very serious diseases, in this case, fungal diseases, which are killers. And if you fail to treat over the first week of a systemic infection when it's growing throughout the body, that puts the patient in a very vulnerable position. So you need to have the right drug. It's not many of them. And then, of course, you need to worry that you don't have an organism which is either naturally resistant or is acquiring a resistance to that drug. And all of these forces make fungal diseases an extremely worrying and major concern as a health burden around the world.
And this was, Neil, just a topic considered by the World Health Organization when a list was published of priority pathogens that we don't have good drugs for and harm an awful lot of people. It would be, I think, helpful when you talk about fungal killers just to describe for people maybe the top 4 that the WHO thinks is most relevant and important, and just a whirlwind tour, if you could, of those pathogens and diseases.
Yes, so in the top 4, there are 15 in the loop, and this list is extremely useful because it formally places fungal diseases and fungi in the mindset of a lot of people who otherwise will have missed them. So it's a really important step forward. The top 4 that they're identifying include Candida albicans, which is the organism I mainly work on, and then a very interesting organism called Candida auris, which I'll return to in a second. A filamentous fungus called Aspergillus fumigatus, which is a mold which grows in compost heaps, but it also is a lung pathogen. And it is a major concern because we're seeing increasing levels of resistance to this organism emerging. And then the 4th one of the top 4, the top priority ones, was Cryptococcus neoformans. And this is a yeast-like organism which grows in the central nervous system. Particularly people who are open to infections, they may often have other infections, including HIV, and they cause meningitis, and that's a very serious disease as well, mainly found in sub-Saharan Africa and other parts of the world, but actually not exclusively. And it's a very worrisome organism with a very high burden, very large numbers. And I may say that overall, we're talking here about the numbers of deaths due to fungal infections as a whole. People are always surprised at these numbers, but shockingly, many more than malaria, many more. Now, malaria has been a success story, and the numbers are going down. But fungal infections which kill are at least 1 million, probably 1.5 million. Some people might put even higher than that. And those numbers are more than malaria, roughly the same or more than tuberculosis and HIV. They're more than numbers of people who are dying each year from prostate cancer and breast cancer, actually. So we're talking a very high number of people who are affected. And in terms of infections overall, we're talking hundreds of millions of mucosal infections of the mucous membranes. We're talking about tens of millions of allergic disease, severe allergic disease, and skin infections. We're talking over 1 billion, maybe 1/5 of the world's population. Now, those top 4, I just wanted to return briefly to Candida auris, because this is a real mystery. The mystery of auris is that for many of us who've been working in the field, like William and myself, for many years, we'd not really heard of Candida auris till about 10 or more years ago. It wasn't in anybody's culture collections. It was virtually unknown as an organism. And then it started to emerge. And what was curious about it, it emerged in different parts of the world at the same time. Now, you might think, well, that's because something like COVID, it spread from one source. That is not the case for auris. It arose, genetically distinct groups of auris strains arose in different parts of the world, which are unrelated to each other. So the same organism popping up in different places, different continents at the same time. Something has changed. There's many hypotheses of what's going on. One of them is even that this might be an example of host jumping, so becoming adapted to a new host because of global warming. That's at least one theory which has been tested now by researchers, that this organism, because of the increasing number of very warm spots, the organism had learned to adapt to warmer temperatures, and become now able to colonize a very warm temperature environment, the human body. So that's unproven at this stage, but it's a possibility. So Candida auris is also very troublesome because it tends to be resistant to a number of drugs, and some of them are to multiple classes of drugs, including the 3 main classes and a limited number of strains, which means we've now got a fully multi-drug resistant fungus, almost impossible to treat. It's also very difficult to get rid of in hospital settings. Now,
maybe
yeah.
Neil, sorry, Neil.
I just wanted, just want to say a little bit about Aspergillus, because
Yes.
I know you also work on
Aspergillus. I was going to ask.
It's a really, really important organism. A lot of work in the UK. There's a major Aspergillus centre in Manchester, which is at the centre of things here. Aspergillus fumigatus is a very common mold in the environment, but it's an increasingly common mold in hospital setting. And one of the things which is of real concern about this fungus is we're seeing now that if you just harvest and select spores from the environment by sucking them out of the air, you find that a significant percentage of those are already resistant to the major class of drugs, the azoles, in fact, the triazoles, which are used to treat them. So that means they come into the clinic, they come into the hospital as a spore which already carries resistance. That's a real concern. And the worry is that this resistance may have emerged because farmers need to treat their cereal crops in particular with fungicides, and they include the same classes of antifungals which are used in the clinic, the azoles. And so the training of resistance occurs in the field, and then the spores get transferred to the patient. The patient then comes in with a very difficult to treat mold, made even more complicated by the fact that there was resistance already. So those are the 4 major ones, and they all have distinct problems associated with treating them.
So yeah, Neil. So thank you. So then, why is it, do you think, there's drug resistance is becoming increasingly important in the public's mind? Funding bodies will often exclude antifungal resistance in calls for new approaches to address drug resistance. So after the case that you've made, large numbers, lethal diseases, few therapeutic options, do funding bodies exclude these organisms from funding calls?
Yes, this is a worry. I do think things are getting better, and that things like the WHO document, the work of people in this field, their advocacy have improved the situation. And we now see the major funders trying to level the playing field much more than they had in the past about working with fungi, and actually also working on AMR. I think the Jim O'Neill report published a few years ago now, but really did a lot of good in pointing out that, if the current trends continue, by the time we get to 2050 and beyond, we may be seeing as many people dying of AMR, inability to treat an infection, as we do with cancer. And so, the amount of money that goes into cancer is enormous compared with anti-infectives. Enormous. I mean, I think the figure I have in my head is for the number of R&D staff working in oncology is estimated to be in about 200,000. In antimicrobials and AMR, it's estimated to be about 600. I think I got these numbers from you at one stage, William, one of your colleagues. And if you think about the antifungal part of that 600, it must be tiny number. But it's really important. 45% of private sector research money goes to oncology. And in terms of infectious disease budgets, those who are funders through government organizations, NIH in the US, MRC, NIHR, Wellcome Trust in the UK, and in the European community countries, it's all about the same. The number always comes out about the same. 2 to 3% of the infectious disease budgets goes to medical mycology, a tiny amount. That is way less than the actual burden of disease. It reflects community size. That's what it doesn't reflect anything else. The research which is done within this community is of high quality. They're as competitive as any others. There's just precious few of them going to be evaluated. The community science has not caught up with the tidal wave of infections which we're increasingly seeing.
Well, so Neil, but how will we address that? So if you were a young researcher, maybe you were going to do a PhD, or you were looking for a career in academic medicine, what advice would you give to people about trying to train, get skills in medical mycology?
Well, I think it's a very attractive area to have a speciality in, because you've got a chance of making a substantial difference. The number of clinically trained people in the UK, as just one small country in global terms, who specialize in mycology, you could count less than the fingers you have on both hands, not many more than the fingers of one hand. Actually, it's a very small community, very hard pressed. But the work that they do is incredibly important, and it's also extremely interesting, both from the fundamental point of view and from the applied point of view. I think there's tremendous interest. The quality of outputs which you see in the literature reflects that interest, and we see incredibly important advances almost monthly about new ways in which we see fungi influencing the human microbiome, and therefore influencing all sorts of other things, how it affects surprising diseases which we thought they were unconnected with, including, for example, alcoholic liver disease and various, even brain function. And in terms of immunological terms, if you look at the ways in which the immune system tackles infections as a whole, you'll find there are more receptors which are targeted against fungi than any other group of pathogens that exist. So the immune system's been coping with fungal infections for a very long time, over evolutionary history. And so it's a very complicated and rewarding area of research to be involved in.
So especially in the post-pandemic era where everything is upside down for one reason or another, what do you think that the next is the model that we've pursued of often very strong and capable investigators, as both you and I benefited from that and trained in that way? Is that a model that's sustainable? Or how would you envisage things being better in the next sort of decade or two to address the challenges that you've so elegantly described, Neil?
Well, I think there are some real opportunities. Obviously, we'll want to build critical mass, and we want to do that as a very collaborative community. We've got to work together. Perhaps there's a bigger onus on us as a community to work together than some of the others. We don't have as many natural patrons of our field, so we need to bring people together creatively to get the people to work across boundaries, to explore interdisciplinary working in a very meaningful way. We need to continue to put pressure on research funders. We need to also take advantage of, what skills we learned in the pandemic about remote working and working together. That will help us a lot. It should help us a lot in training, for example, to be able to take some of our expertise to parts of the world where there are very few local experts, but we can share that expertise much more broadly in the past. And we must keep in mind, actually, that fungi, because they're very flexible organisms, they take advantage of tragedy in many cases. You see them emerging in all sorts of new contexts. And one which you'll have seen a lot as well, William, is COVID-19 itself. So COVID-19 has brought a new wave of infections on the back of the COVID infection. So COVID plus Aspergillus is a real concern. COVID plus Candida is a real concern. And beyond that, COVID or influenza plus Aspergillus, influenza plus Candida are also concerns. We see that fungal infections will also piggyback on the back of other infections to make things a lot worse and much more difficult to diagnose and much more difficult to treat. So we have to keep up to date with all of these emerging pandemics and contribute to that, the global efforts to get on top of them, because we know that some of the most vulnerable patients may not be taken away by the actual viral infection itself. They might be a victim of the co-infection that they have, which in many cases is going to be with the fungus.
Yeah, that's right. So, natural disasters is another one, of course, where that's often Right. So finally, Neil, if you had your time again, would it be Candida albicans and mycology? Has it given you everything that you'd wanted for and hoped for in your career, this field of medical mycology that you've been so intimately involved with for the last X number of years?
Well, I can honestly say that I've never regretted for a second making the choice to work in fungal systems. I think it's been intellectually very engaging. I think the community has been very coherent, and it's been a very welcoming community, and there's a great deal of collegiality in this, partly perhaps because I've often felt a bit like the underdogs looking after one another, but it tends to be very coherent, very exciting. I still think there's a lot to be done. And for those who are looking forward, there's things which have barely even started. I just wanted to mention in that context, the shocking reality that there isn't a single fungal vaccine. Not one. People find that amazing. There's not a single vaccine to any fungus as yet. There's no established immunotherapy as yet. So actually the ability to save patients of serious disease, still we lose often 25%, worst case in this cases, in some diseases, 80, 90% of patients will not survive that infection. There is a huge amount to be done. So my only frustration is that we've not got to the stage yet where we're really providing the types of support that we need to, but I think we're going in the right direction.
Well, Professor Gow, there is the challenge. So thank you for describing the beauty of fungi, the collegiality that you have exhibited and continue to exhibit, the unresolved scientific questions, and the numerous unmet medical needs that will continue to emerge, that drives us all forwards. Neil, thank you for your contributions today and over the course of my own career and the support that you've given me. And long may it continue. Thank you very much.
And vice versa, William. Thank you very much for speaking to me and helping to raise awareness of this really important element of healthcare in not just in the UK, but around the world.
Thank you, Neil. Thank you. Thank you for listening to this episode of Tackling Antimicrobial Resistance, a short NIHR podcast series hosted by me, Professor William Hope. If you have any thoughts or comments on this or any other episode, please contact us via our Twitter channel at NIHR Community. And for more information about the NIHR, you can visit our website, nihr.ac.uk.